This is a brief talk about taurine and could it improve the treatment of cystinosis. Now we know that cysteamine is good, and yesterday and today we've heard a lot of beating up of cysteamine. And those of you who saw the brief history I gave with Dr. Ames' talk know that I did that in 1976. Before that, we knew cysteine was in lysosomes, and people got sick when that happened. And so we thought maybe getting it out would be a good idea. And in Jerry Schneider's lab, I figured out how that worked. This is a nice paper by Dr. Gall's group that just came out. And you can see that compared to the untreated, the patients with cysteamine treated early with adequate doses maintained half their kidney function for as long as that study went on, which is up to 16 years. So it's a good drug. But there are problems with it, and you've heard them described by several speakers, and there's no question that that's true. So what's wrong with cysteamine? Well, one is we have the Fanconi syndrome. And so the children are forming and adults are forming huge amounts of urine, and that causes social distress, but it also wastes small molecules. So if we could control the Fanconi syndrome in cystinosis, we would be making a major advance. But there are other issues. So there's the Fanconi syndrome, there's kidney failure, which is delayed but not averted with cysteamine. There's short stature, which can be treated with growth hormone, but it would be nice if people didn't need to use that. It's an injection, it's expensive, etc. There's muscle weakness, there's hypercholesterolemia. Many of the cysteamine treated cystinosis patients also have to take an anti-lipid drug. And it goes on, male infertility, early aging, vascular calcifications, etc. Now I have to talk to you a little bit about mTOR. And mTOR is the abbreviation for mammalian target of rapamycin. Because the time is limited, I won't bore you with why it's called that, except to say that it was discovered in a bacterium cultured from Easter Island, which in the indigenous language is Rapa Nui. And that compound, that protein in our bodies, which was really discovered and exploited, or at least understood in the past 20 years, does a lot of things like this. I'm sure you can't see that, and I don't want you to read it. But if you look at the two squares in the center and the lower left, gray squares with blue circles, all of those things are controlled by mTOR. It is a conductor of cellular symphony. For 50 years, people have said, oh, the pituitary is the conductor of the endocrine symphony. And that's true. But mTOR is a conductor of intracellular metabolism, and it does that very well, except that, why is it important in cystinosis? Why am I telling you this? And the reason is because, and Dr. Krenantignac's lab first found this, cystinosis is a component of mTOR. You may or may not know that many things in the body that are functional are made up of several different proteins, different genes. They find each other, they combine, and together they do the important work of keeping cells and therefore the entire organism or patient alive. And cystinosin is part of mTOR. So this paper, which was published in Nature, so I have it down at the bottom in red because Nature and Science are the two premier scientific journals. You don't publish there unless your stuff is absolutely true and absolutely earth-shaking. And they reported that cystinosin directs the proximal tubule cells toward growth and proliferation when you don't have cystinosin functioning. And the reason that's true is, and I'll show this in a little detail, but it's more biochemistry than I think we need to talk about today. Cystinosin, as you know, gets cysteine out of lysosomes. And when you don't have that, things go wrong. And what we thought always was that it was the cysteine inside the lysosome. But it turns out that when cysteine gets out of the lysosome, It's then converted to cysteine, and I'll show you that formula in a minute. And that cysteine tells mTOR, okay, everything's fine. I can make specialized cells. I can make proximal tubule cells, and I'll be okay. But when cysteine can't do that, then the mTOR, which senses cysteine as one of the major amino acids that tells it nutrition's okay, goes on the warpath and starts degrading its own proteins with processes called apoptosis and apoptophagy. Um, and it starts degrading things, and that has a lot of bad consequences. I don't think I'll dwell on this today, but you can see there in the right, it's cystinosis deficient, and you see the long green things with purple lollipops at the top are absent in the yellow. Those are the tubule cells that are absorbing things, proteins, from the urine flow and recycling them. And that can't happen when mTOR says, no, I'm not going to make these specialized cells. I'm going to only make the cells it takes for me to survive. And that then provides a direct explanation for the Fanconi syndrome. And you can see that because, as the author said, CTNS, mutated in cystinosis, drives proximal tubule cells toward growth and proliferation and disrupting their function. And therefore, cysteine storage directly redirects the cell not to make proximal tubule cells in the kidney. Many other things, but this is the point I want to make with respect to Fanconi syndrome. And this is my serious take-home lesson from today, and it's not from me. I've quoted all the papers that I read to determine what's going on here. But having been someone who first found how to get cysteine out of cells and thinking that was good, it really isn't the cysteine storage, but it's the cysteine deficiency, which results from cysteine storage in lysosomes, that upsets the apple cart. Now, you may know, and I think it's been said by several speakers this morning, cysteamine reacts with cysteine inside the lysosome to make a new compound called the mixed disulfide of cysteamine and cysteine. And if you look at those two formulas on the left, you'll see cysteine, and we have, it's symmetrical. There's an S and an S. If you break that S, then both of those residual molecules are identical. If you look at the right, you'll see that the mixed disulfide of cysteamine and cysteine only has one cysteine. That's the top half labeled on the right, and the bottom half is cysteamine. But cysteine then only comes out when you have defective cystinosin half as much. So cysteamine does get it out, but it doesn't get it all at once. It has to do many, many cycles to get the amount out. And the other thing is that when we lose the proximal tubule cells, and CRN funded this study of existing cystinosis kidney biopsies, those cells pinch off and form atubular glomeruli. The glomeruli, as I'm sure you all know, are where the plasma is filtered and the urine is formed. And when those cells break and disconnect from the collecting system, then that glomerulus isn't causing any renal function. So integrate all of those cells lost, and you get kidney function in there. In the current day, requires at some point kidney transplant. Now, I'm going to talk briefly about this, though I don't want to get buried in small details. But the cystinosis cell pathology attributable to constitutive mTOR action. In other words, if mTOR has gone on the warpath and says, I got to have other amino acids because I'm starving, even though it's not, but because cysteine can't get there and therefore gives cysteine to mTOR, mTOR does, as I've just said, go on the warpath. And that then means you lose cell specialization, you increase apoptosis, you increase autophagy, you have intermediary metabolism problems, including cholesterol synthesis, which we've already talked about, male infertility, etc., inflammatory problems, and myocyte and osteocyte problems. All of these things are known when mTOR goes on. So in a summary, cysteine reduces the availability of cysteine, that induces catabolism, and then in those cases, we get a diffuse differentiation, a failure to differentiate cells, but increased apoptosis and autophagy. So these are from the inside, what goes wrong in an organism, in cells, when mTOR is not functional. Now we talk about taurine, which is the reason that I guess I'm standing up here. Taurine is an amino acid, 2-aminosulfonic acid. Those of you who remember organic chemistry at all will see that taurine is not a carboxy amino acid. That means it's not proteogenic. So it doesn't form a protein like all the other friendly amino acids like alanine and serine and methionine and cysteine. But it is 70% of the amino acids inside the liver, and most of the cells in the body are taurine. And taurine is an important compound. It functions for osmoregulation. It does a lot of things. But just recently, what has been found is that it corrects some of the things that are wrong in the abnormalities that we see in cystinosis. It's generally recognized as safe, and it's available. So taurine deficiency as a driver of aging. And this is a paper that came in Science 2 years ago. And it's found that in all organisms studied, from worms to mice to monkeys to man, as you age, your taurine concentration goes down. And here's a slide which you may not be able to see, but if you look at the top three, you'll see a mouse, a monkey, and a human, and the decline is very obvious that taurine decreases with age. And in the bottom ones, which again I think you may not be able to see, if you add taurine back, you can increase the lifespan of the experimental animals. Now, this is just something else repeating the parallel between cystinosis and aging. So many of the things that happen to people with cystinosis are a component that we would see in people without cystinosis when they're in their 60s and 70s. But this catalog here that Dr. Gall's group published and others show that it occurs at about age 26 in patients with cystinosis. That's this slide right here. So here's the important thing for today's standpoint, and this was done by Dr. Rezan Topaloglu, who is in Istanbul, and she studied everything in cystinosis that you could find with a very complicated analytical machine and did metabolomic assays. And I, looking through the 240 things that she had quantified, found that taurine was 5% of that found in normal controls, and only and if you give cysteamine, you get some more taurine, but not very much, not enough. So this is her paper from her group, and it's just amazing because this really hasn't been recognized. And as I said yesterday, taurine has been kind of neglected in terms of intermediary metabolism because it forms taurocholate, which is bile acids. It helps you excrete feces, etc. But it really wasn't recognized as an important component of anything to do with inborn errors, specifically cystinosis. So here's why I think, this is my speculation based on the literature, that we have low taurine in cystinosis. There's an enzyme, CSAD, cysteine sulfonic acid decarboxylase, that produces taurine in the body, and its primary substrate is cysteine. And therefore, when you don't have cysteine coming out of the lysosomes from cysteine to cysteine, then CSAD has nothing to work on. So it can't make taurine. And secondly, taurine is a small molecule, and we all know that the Fanconi syndrome wastes and wastes glucose, carnitine, amino acids, calcium, phosphorus, sodium, potassium. So the hose is running, taurine is running out, and there's no input except dietary. And therefore, you have hypotaurinemia. So It has been shown in other conditions, not cystinosis, that taurine supplementation reverses many of the cellular defects which are seen in cystinosis. And I'm not going to read those to you, but I've highlighted them in yellow. And these are some of the things that are known in other conditions that have things that are wrong in cystinosis, like apoptosis, like autophagy, um, like difficulty with muscle formation and bone formation. So again, taurine has been shown to beneficently improve things that are wrong in cystinosis when those things occur in other conditions, but it hasn't been studied in cystinosis. So here we are, the role of taurine in male infertility, another issue in cystinosis that it could help. So my summary, there's a similarity between cystinosis and aging. Taurine decreases with age in many species. It significantly decreased in cystinosis patients with only 5% of normal controls. Supplementation of taurine improves the quality of life and longevity in experimental animals who have, just because of age, lost significant amounts of taurine. And it corrects many of the detrimental effects of chronically active mTOR, and it corrects much of the cystinosis phenotypic elements in vitro and experimental animals. So that's the story, and I want to then just say that for what it's worth, which is not very much, there's a patent on this that the University of Michigan filed. But I do want you to read that if you want to. This was published 3 weeks ago. It's all the details and the references that I didn't take time to go over today. So then I need to say, as a physician and someone who's treated patients with inborn errors and with cystinosis since 1974, you can buy taurine at health food stores. If I were a parent and I had a child with cystinosis, I would say to myself, why don't I go down and buy it and give it to my child and see if it works? And there's nothing illegal about that. It's not paid for by Blue Cross Blue Shield or other health insurers, so it can be done. However, if we do that, we will never know what the correct dose is. We won't know when to start it. My belief is if we start it, if we could do a clinical trial, we would like to start it in the first year of life and then follow that cohort for 5 or 6 years to see, does the Fanconi syndrome go down? Does the excessive urine I showed in that slide not happen anymore? Is our muscles stronger? Our males, this will now take 15 or 14 years from the time we start it to see if it'll correct male infertility. But those are things we can measure. If everybody tries it, or a large substantial segment of people do, then we will sort of get bits and pieces. But the problem is, like other things that have been available for other drugs and tried, it may not give us a clear answer. So with that, thank you for your time.